Welcome to this video on the fundamental unit of life. We will be covering topics such as cell shape and size, cell volume and number, a practical activity to observe onion peel cells, and the concept of division of labor. The basic shape of a eukaryotic cell is spherical, but its final shape is determined by its specific function. Cell shape can be variable, as seen in amoeba, or fixed, as in most plant and animal cells. Cells exhibit diverse shapes like polyhedral, spherical, spindle-shaped, and elongated. Activity 5.1 focuses on making a temporary mount of an onion peel. The materials needed include an onion, a microscope, staining solution like iodine or safranin, and slides. The initial steps involve peeling the skin from the concave part of the onion and transferring it to a watch glass with distilled water. Next, stain the peel with safranin or iodine solution for about 10 minutes, then wash off the excess stain with distilled water. Mount the stained peel on a glass slide using glycerin and a cover slip. Finally, blot the extra glycerin and observe the slide under a compound microscope. Observations will reveal rectangular structures aligned side by side, which are the cells. Each cell has a distinct cell wall and a nucleus at the periphery. A large vacuole and lightly stained cytoplasm are also visible. The presence of a cell wall and large vacuole indicates that the sample is a plant cell. Cell size varies broadly, from tiny bacteria cells measuring 0.2 to 5.0 micrometers to the very large ostrich egg, which is 18 centimeters. Prokaryotic cells are usually smaller, around 1 to 10 micrometers, while eukaryotic cells are typically larger, ranging from 10 to 100 micrometers. Key unit conversions for cell size include 1 micrometer being 10 to the power of -6 meters, 1 nanometer being 10 to the power of -9 meters, and 1 angstrom being 10 to the power of -10 meters. Amoeba proteus is the largest unicellular organism at 60 micrometers, while Mycoplasma gallisepticum is among the smallest cells in multicellular organisms at 0.1 micrometer. The volume of a specific cell type remains relatively constant, regardless of the size of the organism. For example, kidney cells are similarly sized in a horse, cow, or dog. The overall size difference in organs or organisms is due to the number of cells, not the volume of individual cells. While the number of cells is usually indefinite in multicellular organisms, it is fixed in some, like rotifers and nematodes. In nematodes, the cell count is set at hatching, and growth occurs mainly through an increase in cell size. This constant, genetically fixed number of cells is called euteny. In most multicellular organisms, the cell number correlates with the organism's size. Smaller organisms have fewer cells. For instance, human beings are estimated to have about 100 trillion, or 10 to the power of 14, cells. Multicellular organisms exhibit a division of labor. Different parts of the human body perform specialized functions. For example, the stomach lining has various cell types, like mucus cells, chief cells, and parietal cells, each responsible for secreting specific substances vital for digestion. A cell itself also demonstrates division of labor, with specialized components called cell organelles. Each organelle performs a specific task. For example, mitochondria produce energy, chloroplasts synthesize food, and lysosomes clear waste materials. In the next video, we will delve into the structure of the cell, focusing on the plasma membrane, cell wall and membrane, cytoplasm, and nucleus. We will also explore the fluid mosaic model of the plasma membrane.